Welcome back, everybody. I'm so excited to join and welcome our next guest, Dr. Clarice Aiello. Dr. Clarice Aiello is with the QuBiT lab, or the Quantum Biology Tech Lab at UCLA in Los Angeles. Clarice, thank you so much for sharing your day. I think you're traveling. I can't remember if you're coming back from someplace, but thanks so much for joining. Can't wait to hear your talk.
Well, thank you for having me here. Yes, I am traveling. I am in the American Physical Society March meeting. Thanks for having me live and online, even though I'm traveling.
You are online.
And I am super excited to talk to the Montana Quantum folks. Feel free to ask questions during the webinar. And can you relay any questions? Should there be any questions during the webinar? And
I'm certainly can. You bet.
Awesome. Thank you. And so I promise you we're going to get to quantum things very, very soon. So, but I'll start by telling you a little bit about a overlooked, understudied, and many times misunderstood degree of freedom in biology. It has to do with the fact that the whole machinery of the cell, all of those phenomena that you see there plotted in the slide, they seem to be tweakable by weak magnetic fields. And they do so in a way that is consistent with a quantum mechanism underlying how they work. I'm going to tell you how over 40 plus years, this phenomenon has been studied a little bit haphazardly, and how I think we can break clean with studying this phenomena in biology with better quantum instrumentation. And finally, I want to stress that this is not a basic science curiosity. All of those fields that, again, you see plotted in my slides can be improved, can be affected by learning how this degree of freedom of biology works. And even though this is right now science fiction, I do believe that within 1 to 2 decades, we can have what I'm calling endogenous, that is, you don't need to do genetic engineering, non-chemical, everything is electromagnetic, nothing is chemical, non-invasive, cheap, remotely actuated and portable electromagnetic interventions that can actually be positive and that can affect all of those fields below. We're going to get there, and I promise you that we are going to talk about space travel at some point. But first of all, I would like to just thank the people who have been doing this work with me in my group. So I like to call myself a quantum engineer. This means that I build instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get a little bit surprised when I say that I study things that might be going on inside birds and in fact inside all our cells. And that's because I'm a quantum engineer interested in how quantum physics informs biology at the nanoscale. So before I explain where I'm going, I really need to explain to you where I come from very briefly. I am trained in a field known as quantum sensing. The idea is that if you have a quantum object that is put to work as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. In my past, depicted by the leftmost picture, I worked with a technological quantum sensor that, namely an electron spin in diamond, that worked at room temperature as a quantum sensor and under the noisy environment of the diamond lattice and of the noisy environments of the samples that the sensor measured. Some of you might be familiar with this quantum sensor. It's the nitrogen vacancy center diamond. It's basically an electron spin that can be put to use as, for example, a sensor of magnetic fields. But my talk today does not start with the technological quantum sensor. My talk today starts at the center picture, okay? And the center picture talks a little bit about the chemistry. In fact, chemists have known about that for decades, except that they don't call it quantum sensing. But for the chemistry of proteins and of particular electron spins within proteins in solution, there is no doubt that electron spins are behaving as true quantum sensors in the same way that an electron in diamond is a quantum sensor. We're going to talk a little bit about that, about what's known. Okay, and we're going to be bridging the gap between chemistry and physics. And I'll finish, and that's the rightmost picture, I'll finish by showing you correlative, that is not final, but correlative evidence that electron spins within proteins, within cells, might be retaining their quantum character for long enough to be used by the cell as quantum sensors and affect biological function. So that's the basic plan of my talk. And as I mentioned, we're going to start with the chemistry. So the story in chemistry starts because chemists know that weak magnetic fields can alter the final products of a class of chemical reactions that depend on electron spins. So here's how this happens in chemistry. No doubt, this is not controversial at all. There's a chemical reaction happening in a test tube doing its thing, and at some point, the chemical reaction comes to a crossroads. At that point, the chemical reaction is effectively going to look for the spin state of a particular electron spin. Electron spins can be represented by arrows, spin up or spin down, meaning different energies of interaction with a magnetic field. If the particular spin that the chemical reaction cares about is down, the chemical reaction is going to continue through one branch, yielding what I write there as the yellow products. If, on the other hand, the particular electron spin that the chemical reaction cares about is up, the chemical reaction is going to continue through a completely different branch, yielding a macroscopically different product that I depict there by the red products. However, we all know that electron spins are quantum objects. In particular, this means that in the presence of a magnetic field, an electron spin that is down can be put into a superposition of up and down. Sometimes I need to explain what a superposition is to people who are not physicists, and I like to use the analogy of a dimmer switch, right? A magnetic field can put the electron spin in a superposition of up and down, which is a combination of both up and down by a position, by a dimmer switch, by a character that is determined by the magnetic field itself. And we all know that everything that starts quantum dies classical. So after a short time, an electron spin that is described by a dimmer switch is going to be better described by a flip switch. That is, it's either up or down. But for a short time, a weak magnetic field, for example, the blue magnetic field, is going to put the electron spin into a superposition. Now, if you have many molecules undergoing this cycle once, or one molecule undergoing this cycle many, many times, on average, the final products of this particular branch with the presence of the blue magnetic field is going to be a combination of final products of spin only up and spin only down by a combination that is going to be given by the superposition that is itself a function of the external magnetic field. This is to say that a slightly different magnetic field, say the green magnetic field, is going to yield slightly different final products than the products yielded by the blue magnetic field. So this is known to happen in chemistry. There's nothing wrong with that. But usually when we talk about this type of chemical reactions with an implication for biology, this is actually the picture that we talk about. The chemical reactions that are biologically relevant depend not only on one, but on two different electron spins. And two different electron spins can be either in a singlet state, that is one pointing up and one pointing down, or in a triplet state, that is both electron spins pointing in the same direction. Everything else is as I have just described. So I would like to make some comments here. So first of all is that even though the quantumness is short-lived, its consequences are felt macroscopically and at much longer timescales than the timescales of relevance for quantum physics. That is, because everything happens downstream from a crossroads, the final products, when they're formed after quantumness is lost. But in other words, is another way to say that the effect of the quantumness is much longer lived than the quantumness itself, than coherence times. The second, so basically, when the products are formed, the two spins are better described by a flip switch. They are either up, 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 or one up, one down. But the quantumness has, in fact, macroscopic consequences that last for much longer than the quantumness itself. The second point that I would like to make, and I won't explain to you why, but it's well understood. Ask me during the questions. This particular phenomenon is the stuff of weak magnetic fields. This is to say that it's only relatively weak magnetic fields that can alter the final distributions of products of this chemical reaction. Another way of saying that is that for two very large magnetic fields, the distribution of products will not differ. Okay, this is well understood. I'm not explaining to you why, but in other words, magnetic fields that can alter the final distribution of products are relatively weak, on the order of millitesla, on the order of typical hyperfine interactions, that is, strength of interactions between nuclear spins and electron spins. Again, I'm not explaining, but if you plot the magnitude of this magnetosensitivity effect as a function of an external field, you get a very funky behavior that, again, is well understood, whereby the magnetic field sensitivity goes up and down, and as external magnetic fields start being much larger than hyperfine interactions, the magnitude of the effect goes down. It's not monotonically going up or monotonically going down. It goes up and down, and that's very funky and very particular. And finally, I want to point out that here, in the same way as is true for technological quantum sensing, this only works because and while superpositions are being used. Here, superposition is being used as a resource. This is true. This is bona fide quantum sensing, if you will. This is the two-spin version of a simple electron spin magnetometry that people are used to seeing for the technological quantum sensor called the nitrogen vacancy center in Diamond. That works only because and while superpositions last. Okay, so superposition here is necessary for the sensing to take place. So this is true quantum sensing, even though chemists do not call it like this. So those were my three remarks. And again, this has been unambiguously demonstrated at room temperature in liquids, in solids, in gases, and for magnetic fields as tiny as the Earth's, which in turn is orders of magnitude smaller than the magnetic field produced by your cell phone. So this whole shebang goes from chemistry to biology because of birds. As you're going to see, I couldn't care less about birds, but birds really are the reason why I am talking to you right now, because it's because of them that this field migrated from chemistry to biology. Because birds, when they migrate, they have a tiny internal compass, and it's been known for decades that when birds migrate, they use the tiny magnetic field of Earth as a compass, at least as a partial cue. Okay, and people were trying to model by doing experiments with birds how they might be doing so, and the results of the experiments with birds were never matching the theory. Until in 1978, a crazy, at that point, theoretical biophysicist named Klaus Schulten made a hypothesis, which I agree that it's outrageous, and the physicists in the audience will find this hypothesis outrageous. But here's what Klaus Schulten proposed in 1978. He was like, well, were the same type of electron spin-dependent chemical reaction be happening inside a cell, inside a bird, birds and other organisms might sense magnetic fields to the extent that they might sense different physiological concentrations of products that result from such electron spin dependent chemical reaction. Now, the deep implication of that, if birds are truly using this mechanism to function, I'll get to the deep implication in a second. So maybe, you know, and again, it's a bird. This remains to be proven or refuted. Maybe a bird turning right or turning left, maybe they would see different magnetic field lines, and maybe, again, this is a big hypothesis, maybe there's something in their eyes that photomodulates the sensitivity of their vision sense to light, so that maybe birds would tend to go towards the direction where it's brighter for them. Okay, so big hypothesis. So the deep, nobody really knows if that's what's happening with birds, because it's very hard to go from molecules to birds, right? But the deep implication of this hypothesis is as follows, and I have never seen it put quite explicitly as this. If birds are truly using this mechanism to function, the deep implication is that spin superposition, what makes these reactions tick, are maintained inside cells at room temperature for long enough for it to matter. Okay, so what do I mean by long enough? So I told you that this type of electron spin dependent chemical reaction depended on superpositions, and we expect superpositions to die very, very fast, right? The dimmer switch to go to a flip switch very, very fast. So there are way better ways of estimating that, but one can say that an electron can sense a magnetic field if it can learn or process around this magnetic field once before undergoing loss of quantumness, before undergoing decoherence. And again, a back of the envelope calculation shows that for a free electron to learn or process around the magnetic field of the Earth, it takes it about 750 nanoseconds. So if birds are using this type of electron spin dependent chemical reaction to function, the deep implication is that electron spin superpositions need to be maintained inside cells at room temperature for at least 750 nanoseconds, which is a tallish order. Physicists know quantum objects that can maintain their quantumness for at least as long. For example, for naturally occurring nitrogen vacancy centers can retain their quantumness for a couple of microseconds at room temperature, but the nitrogen vacancy center is a little bit special. So for an electron spin within a protein to actually survive as a quantum object for that long, this remains a more or less tallish order, right? So this brings me to the next point in my talk, which is to say that this hypothesis, which remains a little bit outrageous, has never been unambiguously verified or refuted by any experiment. There is no doubt that things are quantum, and I'm going to show to you a little bit in a second. There's no doubt that things are quantum for that long for proteins in solution. However, nobody has unambiguously proven or refuted that this quantumness translates, this quantumness is maintained inside, say, single cells. Okay? So if I needed to give a proper title to my talk, it would be this one. So if they are maintained inside cells for long enough to work as a resource, is it possible that we harness those electron spin superpositions for function? Is it possible that we learn how to tweak those electron spin superpositions in the same way that we tweak quantum states, for example, from technological quantum sensors? Because in the end, birds fly, right? And the only standing theory on how they do so is by this type of electron spin dependent chemical reaction. So Klaus Schulten made the proposal in 1978. Biological experimental biophysicists started looking for molecules that can sustain this type of electron spin dependent chemical reaction. And in the 2000s, the first animal, because birds, the first animal protein that could sustain this type of electron spin dependent chemical reaction was found. This protein is depicted there in the screen. This protein is called a cryptochrome. Now, cryptochrome is present in the eyes of birds, in the antennas of migrating butterflies, but surprise, surprise, cryptochrome is present in all and expressed in all our human cells because it has circadian rhythm regulation functions. Cryptochrome actually has a chromophore. It's a flavin chromophore, which means that once you shine light onto this molecule, it absorbs this light, gets excited, and then it de-excites in emitting light. When actually you are jet lagged and people say, oh, look at blue light, one of the things that blue light does is excite the cryptochrome molecules that can actually absorb blue light. And over the course of 20 plus years, people have really shown that magnetosensing might be related to cryptochrome, unfortunately, at very disconnected length scales, at two length scales that are very disconnected. At chemical length scales for cryptochromes in solution, there is no doubt that electron spins in cryptochrome are being used as quantum-based, quantum-enhanced magnetic field sensors. And here's how that works. Again, no doubt about that. I told you that in the chemical reactions that we care about, oops, is there?
Is it delayed or?
I don't. Can you hear me?
I can hear
Hello?
you. Yes. Yeah.
Awesome.
I can hear you.
Okay.
Yes.
I'm not sure what went on here. Okay, sorry for that.
Okay, might have just been a glitch. So I'm mesmerized by your talk.
Okay. So electron spins in cryptochrome are magnetic field sensors in a quantum sense. So I told you that for the biologically relevant chemical reactions, they depend on two electron spins. One electron spin comes from an electron in the flavin chromophore, and the other electron spin in the most well-accepted model comes from a nearby tryptophan. And the way that you actually study these spins in the cryptochrome is by shining light at a vial with those molecules. You shine light, you excite those molecules, and then they de-excite emitting light, and this light you can collect with a single photodetector. Very much similarly to other quantum sensors of magnetic fields, this molecule also has a fantastic property, which is the fact that it has what we call spin-dependent fluorescence intensity, and that's quite remarkable. This means that just by looking at how strong a fluorescence signal this molecule is emitting, you can actually trace back to which electron spins this signal comes from. And here's how this works in the photophysics. And again, if you're familiar with the photophysics of nitrogen vacancy centers, you're going to see that this is very similar. So here I depict a flavin electron spin. Flavin is abbreviated as FAD. It's in its ground state. There's also an excited state that you can access by shining light at this molecule. This electron spin is going to absorb light, get excited, and then most time it's going to emit light by fluorescing down. However, there is a competing pathway here in this photophysics. Sometimes upon photon absorption, this electron spin is going to undergo a process that chemists call intersystem crossing. In this process, this electron is going to meet with our second electron of interest coming from the tryptophan, and in the presence of both an external magnetic field and an internal magnetic field caused by a nearby nuclear spins, also referred to as a hyperfine interaction, those two electrons are going to be put into a superposition of singlet and triplet state. There is not a lot of difference of energy between those two states, but after some time, this electron spin from the flavin is going to want to relax down to the ground state, which it will do, but it will not emit light. It can decay back to the ground state via a vibrationally assisted process. And there are two possibilities. Either this flavin electron decays back while the two electrons are in a singlet state, or it decays back when the two electrons are in a triplet state. But as depicted by the strength of my arrow there, it's way more likely for the electron to decay back if the two spins are in a singlet state. So there are three competing processes there, one that emits light and two that do not emit light. So if you have one molecule, one cryptochrome molecule undergoing the cycle many, many times, or many cryptochrome molecules undergoing the cycle once, on average, because if you are in a singlet state, you have a higher chance of being brought back to this light emitting process again. On average, if you are in a singlet state, this molecule is going to emit a little bit more light. It's going to emit about up to 5% more fluorescence. So just by looking at fluorescence intensity, you have access to the electron spin state. You can do a true quantum state readout if you have a very good way of counting photons, okay? But the idea here is that this fluorescence intensity is tiny, but it's measurable. So if you can count photons coming from this molecule very, very well, you can do a true quantum state measurement. And again, this quantum state is going to be a reflection of the magnetic fields that this molecule sees, so that you have, in other words, a true magnetic field sensor and access to this magnetic field sensor, experimental access via fluorescence intensity. And then there's no doubt that things are quantum in this way, and actually people have been able to measure fluorescence intensity as a function of magnetic field. So in this plot, you see fluorescence intensity for a vial of cryptochromes as a function of the time during which this vial was excited with blue light. As you can see, this fluorescence is going down. This is called bleaching. You're killing the chromophore. But as the researchers pulsed a tiny magnetic field on and off as this fluorescence was being killed by the laser, they saw a slight difference in fluorescence intensity for the reasons that I have just outlined, because cryptochrome spins are working as true magnetic field sensors. There's no doubt that this is what happens for cryptochrome at room temperature and in solution. However, the next level of evidence for this mechanism comes from organisms such as birds, flies, planaria, plates of cells that respond to magnetic fields in a way that is consistent with this type of electron spin dependent chemical reaction, being alive inside their cells for long enough. And as I mentioned, this has never been unambiguously verified or refuted. So can we do better? Is there a way to populate something in between proteins in solution and big birds, right? And in fact, we think we can do better. Which brings me to the third part of my talk, given by this rightmost picture there, because this is not a talk about birds and nor a talk only about cryptochrome. All of those processes that you see there were shown to be tweakable by magnetic fields in a way that is consistent to this chemical process that I have just described. And again, cryptochromes have been shown to be responsive, that have been shown to be involved, but there are some processes that do not necessarily involve cryptochrome. They involve other proteins, they involve other radicals that might be active. And here's a big summary of papers that we have been published in this space. And you have, those are my favorites, but sometimes people get what I'm talking about when I show them this picture there. This is a picture that comes with a story. I have a friend who's a precision measurement physicist, and his name is Peter Furlinger. He's at the TU Munich. And usually what he does is he builds chambers called hypermagnetic chambers that shield very well the external magnetic field. So inside his chambers, he has single nanotesla of residual field. And once a biologist friend asked him to grow things instead inside of his very good hypermagnetic chamber. And he grew tadpoles for 2 days because that's as long as he could go without asking for a bio permit. He grew tadpoles for 2 days inside the chamber with a mimicked magnetic field of the Earth inside, and the tadpoles looked normal. If instead he grew the tadpoles under only the 2 nanotesla DC field, he saw that about 35% of the tadpoles were malformed, were non-viable after 2 days. So let's recap. You don't even need to add a magnetic field. It suffices to subtract the tiny magnetic field of the Earth, and you see biology working a little bit crazy. So this opens up all sorts of like Pandora box type questions. For example, the magnetic field of Mars is about 1 divided by 50 the magnetic field of the Earth. Can you space colonize Mars? Can you reproduce in Mars? Can you space farm in Mars, right? If it seems that life is adapted to the tiny magnetic field of the Earth. And nobody really knows what's going on inside the tadpoles. But again, if the tadpoles are sensing the magnetic field of the Earth, or its lack, due to this type of quantum mechanism, there is no doubt that they do this using this type. Oops.
Oh, I can still hear you.
Weird. You can hear me.
Advancing?
Can you hear me?
I can hear you. Yep.
Okay, awesome.
There's a little glitch here.
Sorry, glitch again.
Yeah.
So if the tadpoles are sensing the magnetic field of the Earth using this quantum enhanced mechanism, the profound implication is that electron spin coherences are alive for at least 750 nanoseconds. So I just want to mention a point that the magnetic field of the Earth is very, very tiny. If you want a cell or an electron spin inside a cell to sense a magnetic field such as that produced by your cell phone, which is on the millitesla strength order, this would only require coherence times of about 50 nanoseconds. Okay, so sensing the magnetic field of the Earth is especially difficult because it would require 750 nanoseconds of coherence or longer, but maybe it's way easier for electron spins to sense the magnetic field, say, of your cell phone, on the order of your cell phone, which I believe, and this is a hypothesis, is what's happening in some applications that people see. So people for many, many decades have been finding relatively weak, on the order of millitesla magnetic fields that do stuff, okay, in a completely haphazardly way. For example, and this is this picture to the left here, there's this company called Novocure that found a weak magnetic field that actually can reduce the size of brain cancer. Okay, and they have this weak field sort of electrode here. The company itself is valued at $4 billion, and they don't really know how it works. And that's those 4 orange blobs there. They say it's a combination of 4 factors. It's a disruption of mitosis, changes in mitosis rates of the tumor. It has to do with cytoskeleton rearrangements. It has to do with DNA repair, and it has to do with ROS, the reactive oxygen species state, oxidative stress state of the cells. And those 4 things have been shown by our group and by other groups to be tweakable by weak magnetic fields in a way that is consistent with this type of electron spin dependent chemical reaction. To the right here, you see 3 pictures from Alfredo Obregon from the National University of Singapore, and he has not 1, not 2, but 3 different startups that found magnetic fields to do different things that go from reduction of breast cancer to muscle regeneration to increase in the proliferation rate of lab-grown meat. Okay, so again, for 40 years, people have been finding magnetic fields that do stuff, and they have been exploring this huge phase space of magnetic field frequencies, strength, direction, completely haphazardly. I liken it to being in a haystack, grabbing a little bit of hay and finding a needle. So there's no predictive power on which magnetic fields do what. So I think we can break clean with this way of taking data. I hope we can get data that is systematic and predictive power. And to do so, I propose that we gather 4 ingredients. I propose that we gather a microscope, a magnetic field, a magnet, synchronize everything down to the nanosecond, and grab a very good photo detector, because as I said, by tracking fluorescence intensities, you can track electron spin state. You put those 4 ingredients together, you can call it a glorified microscope. I call it a quantum instrument. This type of quantum instrument, I'm going to show you where we are, is going to yield what I hope will be causal data that, added to simulation systematics, is going to yield what I'm tentatively calling the code book on how to control spins in biology for function. And again, even though this is a big science project, we cannot lose the technological goal from our side. It's completely science fiction, but I hope that within 2 decades, we have, say, an app in your cell phone where you can go and then you go there and say wound healing. And then the app in your phone produces the exact magnetic field frequency and intensity that you need to do wound healing. And then you go like this. Completely science fiction, but we cannot forget that this has a technological goal in mind. Let me tell you where we are in our big goal of finding this deterministic code book. I'm going to show you the progress that we've been making onto the building of 2 microscopes with those 4 ingredients. That's what the blueprints of our microscopes look like. Okay, where we have we will have the capability of doing both spin measurements and also a regular cellular microscopy. We're building 2 such microscopes, one which is a lattice light sheet with a microscope with a magnet, and the other one which is a total internal reflection, a microscope with a magnet. We're not there yet. Those instruments are not ready to take data. But let me tell you very briefly where we are right now. While we wait for those big instruments to go live, we have been taken more correlative data of weak magnetic field effects in cells. So for the physicists in the audience, this is what it looks like. This box to the left is an incubator. This is where cells live. And we can shove things inside this incubator, such as this commercially available hypomagnetic field chamber. And also we developed active coils that are water-cooled and that do not cause an increase in the temperature in the samples. And we can put things, grow cells inside either both the hypomagnetic environment or these active added magnetic field. And here's some effects that we see and that corroborate existing literature. Okay, so what we see, for example, is that a very modest added magnetic field on the order of half that produced by your cell phone can indeed produce changes in the cytoskeleton of the cell. The cytoskeleton of the cell is composed of mainly 2 different types of fibers, actin and microtubules. Actins are depicted in green to the left, and microtubules are depicted in pink to the right. And we see that a modest magnetic field altered the shape of actin filaments, alter the shape of how much the microtubules they aggregate. Okay, so this is in agreement with previously existing literature. We also see that. In fact, we can do experiments of how easy it is for these microtubules to form after we disrupt this network using a chemical. And we see that in the presence of magnetic fields, this network of microtubules is after a couple of hours is not formed back. You can really alter those macroscopic parameters in the cell as a function of very weak magnetic fields. We can also see, as per previous literature, that you can change the oxidative state of the cells by weak magnetic fields. And all of this is, again, consistent with this type of electron spin dependent chemical reaction being active inside cells for long enough to sense this type of super weak magnetic field. So this brings me to the end of this very brief talk. I'm going to end in the way that I always end, which is by saying, may the quantum be with you.
I love that, Clarice. Thank you so much. There was so much in here. I don't know how many requests for these slides, or just at least the recording will be made available. This is sort of a whole masterclass deep dive semester, at least, of introduction to what you're doing. Before you go, I know we're a little bit over time, but Clarice, what's next? I mean, you mentioned some of the things that you're doing in terms of building this. What are some of the most near-term things that your team needs, and what you're seeing as kind of exciting opportunities, or things that you know?
So data on these magnetic field effects in biology have been taken for over 40 years, and the needle never seems to move, right? So we maintain that one thing that might make the needle move is indeed when those quantum instruments go live, right? So in order for quantum biology in general, and this flavor of quantum biology in particular, to start being more taken seriously by the scientific community, we think it's important to unambiguously show or refute whether electron spin superpositions can be alive inside cells for long enough for it to matter, right? I think nobody doubts that things might remain quantum for, say, picoseconds, but in order for those electron spins to sense magnetic fields, we need quantumness times of on the order of nanoseconds. So we need to unambiguously prove or refute whether that's the case, which is the same as we need saying that we need to take bona fide true quantum measurements inside proteins, inside cells, right? And for that, we need the quantum instruments. I think that this will make the quantum people believe in this partial data that is available, right? The chemistry data that is no doubt that things are quantum, and the correlative data coming from biology. So I think there is a lot of room for the quantum physicists to enter this field, because we are used to developing instrumentation. So our near-term and not near-term goals, we can continue to take the sort of correlative data for decades, and that's not going to make a difference. We need to make this shift by finishing to build those quantum instruments. So that's what's really, really needed. And that's going to take as long as it takes to build any quantum instrument, which is 2 to 3 years only in the building.
Yeah, absolutely. Absolutely. Well, Clarice, thank you so much. I'm going to check real quick. There may be a couple of questions. And again, guys, I know, thank you for staying longer. We have another panel starting in 2 minutes. I know people may need a bio break. Are you using, here's a question in the Q&A. Are you using silicon photonics PIC for detection of electron spin dependent quantum fluorescence?
So we're using single photon detectors, right? Single photon detectors and a camera, either in like a single pixel, or we have access to a collaborator of ours who has like a SPAD array, a single photon detector array, which is effectively a camera of single photon detectors.
Yeah. Okay. Cool. Thank you. Next question: does your lab partner with startups?
Yes, in fact, I didn't want to mention this, but we left UCLA and we're right now our own startup.
That's right.
And
it's in Los Angeles, right? Okay, yes, good.
Yes, we're still in Los Angeles, and we are looking for partners to help us develop the technology. Yes, feel free to reach me. I'll put my email here in the chat.
In the chat, and your LinkedIn is there, and I have it too. If anybody misses it out, I can share it as well. But thank you.
And yes, so anyone who has a technology that we could use in our big instruments, I would be super happy to talk to you.
Clarice, I thank you so much time. I'm going to jump over to this other panel that's starting now. It's safe
Thank
travel.
you for having me. Thank
Guys,
you.
great
Take care,
day.
everyone. Bye-bye.
Bye, everyone. Heading over to the next panel right now.